Okay, in this video we're going to talk a little bit about a very common and very useful structural analysis tool used by organic chemists called nuclear magnetic resonance spectroscopy, also known as NMR. I have a introduction slide here just to give you an idea about what we're going to be talking about. The first thing we're going to learn about is a little bit of basics about the output for what you get in NMR, what the spectra looks like for carbon and proton NMR, and what to look for, and what the axes are like, and things like that. Once we've done that, we'll talk about one of the key skills in being able to be successful with proton and carbon NMR, and that's the ability to be able to identify different numbers of protons and carbon atoms in a molecule, because each of those is going to lead to a different signal. We'll also talk about the concept of chemical shift, which is related to the atom's environment within a molecule, so the hydrogen's environment in terms of proton NMR. Basically, it's related to what atoms the hydrogen is next to. We'll also talk about the concept of integration of peaks, and in this case, integration means what it means in the usual sense, like you'd usually integrate under a peak to find the under a curve to find its area. This is kind of similar. We'll do integration of a peak to figure out how many hydrogens are in that peak. And then we'll talk about one of the things that's of in general most problem to most students studying this stuff, and that's something called spin-spin splitting. One of the things that makes proton NMR so powerful and relatively confusing is the fact that hydrogens on adjacent carbons interact with each other, and when they interact with each other they give distinct what's called splitting patterns, and an ability to understand what those mean is paramount to understanding what this NMR spectroscopy is all about in terms of how you can figure out structures. And the last thing we'll just go through is the fact that after going through these first 5 bullet points, you'll see that figuring out NMR spectra really is not all that difficult. So let's start off here. This is basically a generic spectra that you get in proton NMR. There are no peaks or signals shown on here, but I just have the axes shown and a couple of key words so you can see exactly what's going on. Notice the x-axis goes from 0 to 10, sometimes it'll go to 12. Units here, ppm, parts per million. You'll also see this axis labeled as chemical shift, or with the lowercase delta. You'll see the intensity being the y-axis. The higher peaks tend to be more intense. The place where your hydrogens appear, or the peaks due to your hydrogens appear, can be anywhere between 0 and 10, and the further to the left they are, the further so-called downfield they are. This means the hydrogens are closer to something electronegative, whether that's oxygen or hydrogen, oxygen or nitrogen or sulfur or chlorine or something like that. The further to the right, the further to 0 they are, then that means the furthest away, or the further away from something electronegative they are. In fact, this peak at 0, you'll see this in virtually every proton NMR spectrum. This is an internal standard called tetramethylsilane. It's always put in here so you can set this to 0, and then everything is then has a chemical shift relative to that 0. Okay, so like I said, it's an internal standard. So that's the basics of an NMR spectrum, particularly for proton. Now, one of the things I said we were going to look at was the ability to tell how many different types of hydrogens and carbons you have in a molecule. So let's just look at molecule A here. One of the key things you want to make sure you have an understanding of when you're looking at this is the concept of symmetry. If a molecule has symmetry, then you're going to find a lot of the atoms are the same. So you want to look for planes of symmetry, because that's seriously going to cut down on the number of different types of atoms you have. So if we look at this one, everybody will be able to see there's a plane of symmetry right through the middle. And there's also a plane of symmetry this way as well. Now what that means is the two alkene carbons are exactly the same. This methyl group and this methyl group are the same. This and this are the same. In fact, all 4 are similar. Well, not similar, they're all exactly the same. So what that means is we'd see 2 carbon signals. Now if all 4 methyl groups are the same, then that must mean all the hydrogens are the same, so we'd only see 1 hydrogen signal. When we move on to compound B, You remember, look for that plane of symmetry, and there is one right through here. So that means everything on this side is the same as everything on this side. And if we look at carbons first, we got carbon 1. We got carbon 2, two of which are the same there. Carbon 3. Alright, so we got 1, 2, 3. Here we got carbon 4. And then we got carbon 5. And both of those are the same, those two methyl groups. So we've got 5 carbon signals. And when we look at our hydrogens, we'll just get rid of this stuff. When we look at our hydrogens, we've got the two methyl groups. We already said those are the same, so that's one type of hydrogen. These two CH2s are the same. These two CH2s are the same, but they're different to each other. So these guys, we'll call this type 2 if this is type 1. These are different to type 3. Alright, simply because what they're attached to on the outskirts of the ring here is different. Carbonyl group, 2 methyls. Alright, let's speed up for the others. This guy here, planar symmetry right down the middle. These two methyl groups are the same, the carbon in the middle is different, so therefore 2 carbon signals. If both the metals are the same, there's only one type of hydrogen. When we move on to here, planar symmetry gets a little bit more difficult to define, but we do have some groups that are all the same. These 3 CH3s are all the same. So let's look at carbons. We've got one type of carbon here. All those connected to this carbon, which is different to all those, so that's type 1. This is type 2. The carbonyl is type 3. And that methyl is type 4. So when we look at the hydrogens, all these 3 are the same, so that's type 1. There's only one other type of hydrogen, so that is type 2. Now when we do this guy, we've got symmetry here, and we've got symmetry here. So the two methyls are the same. This carbon and this carbon are also the same. This carbon and this carbon are the same. So if we call the methyls type 1, that carbon type 2, these are type 3. This is also type 2. These are also type 3. So therefore we've got 3 carbons. And when we look at the hydrogens, the two methyls are the same. These 2 sets of hydrogens are the same, so we have 2 sets of hydrogens. So when we come to this guy, it's the same thing again. We've got symmetry. 2 methyls are the same. We've got this carbon, and then we've got these 2 CH2s. The 2 CH2s are one type, type 2, type 3. So there's 3 types of carbons here. The 2 CH2s are exactly the same, so that there's one type of hydrogen, another one for the methyls, so 2 types of hydrogens. Now when we look at this molecule down here, it looks more complicated. Because we can freely rotate around this carbon-carbon bond, we can actually say, it may not look it, but we can say there's a planar symmetry throughout the middle here. So what that means is when we look at simply the carbons, we've got type 1 here. We've got type 2 here. We've got type 3 here. We've got type 4 here. We've got type 5 here. We've got type 6 and 7. Notice that 6 and 7 are not the same. One's connected to the ring and one's connected to an ethyl, so they're different. Type 8 and type 9, so that's 9 carbons. And when we look at our proton signals, we'll find it's somewhat similar. Type 1, type 2, type 3, type 4, type 5, type 6, type 7. So usually when the carbons are different, the hydrogens on those carbons are also different. Okay, little bit of basics here. When you're looking at a proton spectrum, depending on the functional groups they're connected to, the hydrogens or the protons usually occur within specific regions of the NMR spectrum. However, when you've got a hydrogen between 2 different functional groups, it could be somewhere other than one of these regions shown. The most important region, I think, for you guys to see is this one right here, because this one virtually never changes. Aromatic ring hydrogens are always going to be between about 6.5 and 8 ppm. And yes, it's true, the hydrogen is not connected to something electronegative, but for reasons involving the resonance and delocalization within the ring, these hydrogens are particularly downfield. You'll be given data tables like this on your quizzes and exams, so you can use these as reference guides. These are just somewhat specific chemical shifts for hydrogens when there's just one functional group involved. And it's exactly the same thing for when you're looking at carbon NMR spectrum. When you look at this guy right here, this shows the kind of chemical shift you'd expect to see in a carbon NMR depending on the functional group involved. So there's a whole bunch of different variety for whatever that functional group is. And when we look at this molecule down here, this is just butanone. There are 1, 2, 3, 4 carbons. They're all color coordinated with the spectral lines here. Notice the carbonyl carbon, the one that's most downfield, is what you would expect. It's closest to the oxygen. And using that theory, you'd expect carbon 1 and 3 to have essentially the same chemical shift because they're equidistant from that carbonyl. And notice they're not exactly that. Carbon 3 is more downfield than carbon 1. And that's because that's a CH2 and that's a CH3. When you have a situation where you have groups like this that are equidistant, if you had, say, a CH3, a CH, and a CH2, the CH will be furthest downfield and the CH2 than the CH3. It's always that way. And that's why it's pretty much always CH3 groups that end up being the furthest upfield, just like this one. Alright, the CH3 is on the end of the chain, it's furthest from the carbonyl, therefore it's the most upfield. And we can say exactly the same thing when we're looking at a proton spectrum. When we look at this guy, this is methyl acetate, you'll see there are 2 types of hydrogens here. Therefore we would expect 2 signals, which is what we see. These hydrogens are closest to an oxygen, or closer to an oxygen than these, therefore they appear more downfield. Note the presence of the TMS that I told you about before. When we look at the carbon NMR spectrum of the same molecule, notice there are 1, 2, 3 different carbons, therefore we should expect 3 signals, and actually that is exactly what we see. The carbonyl carbon, as again, is furthest downfield. This CH3 is closer to the oxygen, therefore it's more downfield than this CH3, which is farther away from the oxygen. Okay, couple of things that I want to illustrate on this spectrum. First thing I want to illustrate is the concept of integration. Notice on this proton spectrum there's this funky line here, as well as these weird looking signals. There are 2 hydrogens, or 2 types of hydrogens in this molecule because there are 2 signals. And one of the things we want to be able to do, unless the number of hydrogens is given above the peak, which it sometimes is, we want to be able to figure out, well, how many hydrogens correspond to this signal? And one way to do it is to use this curve that's given there. Notice that on each part of these curves, this is the curve bit, on each part of the curve there is a straight line portion. There's a straight line portion here, and a straight line portion here. And you want to find a way to measure the distance between those two. You can just take a ruler or something and do that. So that distance there. And then we're going to do the same thing between the straight line here and the straight line here. So you're going to find there's a distance there and a distance there, and those exist in a particular whole number ratio to each other. In this case, it's 2 to 3. Therefore, what this means is there's 2 hydrogens here and 3 hydrogens here, or at least 3 hydrogens compared to 2. This could be 6 and 4, or 9 and 6, or something like that. So these exist in a ratio of 3 to 2, and note that we have 10 hydrogens here. Okay. We have 10 hydrogens and only 2 signals, so that tells us we have a lot of symmetry. And if we have 3 hydrogens here and 2 here, 5 total, that probably means we have 2 sets of 5 to give this 10. Remember we said there was a lot of symmetry? Well, there are only 2 possible functional groups that can have 1 single oxygen and this formula. When you look at the degrees of unsaturation, C4H10 is actually completely unsaturated. So the only possibilities for this are either an alcohol or an ether. Now for an alcohol you always have that OH peak, which first of all would require a peak with an integration of 1, which we don't have, and the alcohol peak always appears as kind of a broad peak all by itself. Kind of broad, B for broad, alright, which we certainly don't see here, and there's nothing that has an integration of 1 either. So therefore we must have an ether. And because of the fact we only have 2 signals here, it's probably a symmetrical ether. The question that we've got to figure out then is, what exactly is this comprised of? We know it's a symmetrical ether, but we don't know exactly what's what. This is where we're going to go through the spin-spin splitting, so don't get freaked out when I start talking about it. It's not that difficult. Let's look at the first peak. This is a signal that's split into 4 peaks. This is known as a quartet. All right. The naming convention goes singlet for 1, doublet for 2, triplet for 3, quartet for 4, quintet for 5, sextet for 6, septet for 7, and so on and so on. So this is a quartet, just as this is a triplet. Now, what exactly does this mean? Well, this is a quartet because it's split into 4 peaks. This is a triplet because it's split into 3. We're going to apply something called the n+1 rule to figure out what is going on here. n+1 for the case of this signal here is 4 because that's a quartet. Therefore, in this case, n is equal to 3. Now, it's important that you understand what exactly this n equals 3 means. It means that these 2 hydrogens here, with an n value of 3, are seeing next to them 3 other hydrogens. So it doesn't mean that here there are 3 hydrogens. It means next to here are 3 hydrogens. And what's the only carbon group you can come up with that has 3 hydrogens on it? Well, it's a CH3. So what that means is for these 2 hydrogens, in whatever form they may be, probably a CH2, there is a CH3 next to them. CH3 next to this guy. All right. And when we do the same thing here, n+1 in this case equals 3. Therefore, n must equal 2. Therefore, these 3 hydrogens are next to 2 hydrogens. Therefore, if this is a peak representing 3 hydrogens, probably a CH3, and it's a triplet, it means it's next to 2 hydrogens, which is probably a CH2. All right. So what we've got here is we have this 2 hydrogen peak, this CH2. Its multiplicity or its shape is telling us it's next to a CH3. And this triplet here, which is a CH3, is telling us it's a triplet, therefore it's next to a CH2. So we actually have the same thing being told 2 different ways. CH2 next to a CH3, CH3 next to a CH2. Therefore, what we're having is a CH2 next to a CH3. Now, before we finish on this, there's one more thing to talk about. You remember what we said before about the furthest downfield you are, the closer you are to something electronegative? Well, that's exactly true here. The CH2 must be closer to the oxygen because it's further downfield, and so that's why we can say this is the structure. All right? Okay. Let's just do one more of these. We have something similar going on here. We have this formula. When we take out these 2 bromines, they get replaced with hydrogens, which gives us C3H8, which again means there's no unsaturation, so there's no double bonds or rings or anything like that. So we have these 6 hydrogens and only 2 signals. So again, we have the possibility of some symmetry. Now let's look at the shapes of these again. We have a triplet, and we have in this case something else. 1, 2, 3, 4, 5. We have a quintet. All right, 5 peaks. Now let's look at our integrations. All right, there's one, there's the other. Let's extend that line over a bit. So we're going to say we have this distance here, and then when we do the same thing and put that line there, and then we measure these 2, what we get is the fact that this peak is actually twice as big as this one. 4 to 2 or 2 to 1, or however you want to look at it. When I come up with a ratio of 4 to 2 though, notice that 4 plus 2 is 6 hydrogens, so it's probably the 6 in the molecule. But then how do we figure out what gives rise to a quintet? We've seen what gives rise to a triplet, so we know that these 4 hydrogens must be next to a CH2. So a quintet is hopefully going to say the opposite of this. A quintet means our n is 4, so therefore that means something, this 2 hydrogens, a CH2 next to a CH4. Well, that can't be a possibility. We can't have anything next to a CH4. But what we can have is two equivalent things that add up to 4 hydrogens, which the simplest way to figure that out is 2 CH2s. So what we're saying then is we have a CH2 here next to 2 others. And then we also have these 2 CH2s next to a CH2 themselves. So what it looks like is we have something like 3 CH2s all in a row. But notice that the 4 hydrogens here are more downfield than these two, which implies these 2 CH2s are closer to something electronegative, and of course that something electronegative is bromine. Alright, this carbon already has 4 bonds to it, the outer ones do not, so we put the bromines on, and what we get is that. So what that means is these hydrogens and these hydrogens should be furthest downfield, which is exactly what they are. Both of these would appear as a triplet because they're next to this CH2. And this CH2 would see 2 hydrogens here, both of which are the same, so they'd add up to 4, giving us that quintet. Alright, one more thing before I finish here. I stressed the importance of remembering the chemical shift of aromatic rings before, in terms of protons, and there's one more thing I want to talk about that. Usually when you're looking at a when you're looking at a benzene ring, in particular a mono-substituted benzene ring, you got to be careful you don't try to read too much into it, because what you tend to get is just kind of a mess in terms of the hydrogens. The logic that we've been through would suggest a much more simple splitting pattern than what we see here. This is obviously much more complicated than what you would expect. And that's primarily due to the delocalization of the electrons around the ring, making for a much more complicated system. So when you have that mono-substituted benzene ring, you can't really tell anything or be expected to figure anything out from this other than the fact it's a 5, it's a mono-substituted ring. However, in some cases, in one particular case, you will have a situation where that splitting is very different, like we have here. This looks like we have 2 2s here, so that implies 2 doublets. The doublets look kind of funky, but they are doublets. This is the case you'll see only when you have this sort of substitution going on the ring. When you have a para-disubstituted benzene ring where X and Y are different. Let me draw on these 4 hydrogens so you can see what I'm talking about. This hydrogen are the same. This hydrogen are the same, but they're not the same as each other, assuming Y and X are different, which in most cases they will be. But let's think about the splitting. If we call these A and these B, and you were to be asked, well, what do you think the shape of HA would be? What would the splitting be? What would its multiplicity be? Well, we apply the n+1 rule, where n is the number of hydrogens on adjacent carbons, and we say, well, okay, HA is on that carbon. On this adjacent carbon, there are, well, there's actually no hydrogens. On this adjacent carbon, there's 1 hydrogen, so n is 1, so n+1 is 2. So therefore HA would appear as a doublet. And because HA is the same on here as here, you actually see that twice with that number 2 there. So you get an integration of 2. And when you look at HB, it's exactly the same thing. There's no hydrogens here, but there is a hydrogen on that adjacent carbon, so that would also appear as a doublet with an n value of 1. So we get that one here. So whenever you have that para-disubstituted benzene ring, you'll always see some splitting like this. In fact, in some cases you might even see a pair of doublets that look suspiciously like a quartet, and you've got to be really careful with that. You'll see an integration that looks like this, which clearly tells you that there are 2 doublets there, or it's not just 1 quartet. Or you'll see something like this in terms of the integration. You'll see something like this, alright, and so there's actually a break there, so that implies 2 different types of hydrogen. So that's an important thing to remember about protons on a benzene ring, and that is it. Thank you.